Welcome everyone to this week's edition of the Extracellular Vesicle Club. We're actually just a few days out from ICEV 2023. So many of you were there. I see a lot of familiar names in the room here. And I hope you enjoyed ICEV 2023. I certainly did. We were just talking about some of the new features that were shown there, but EV Club doesn't stop. So we're back in action this Wednesday, and we'd like to welcome you all to what I think is going to be a very interesting session. Because we're going to be looking deep inside the cell, what's going on in the cell as we start to have exosome biogenesis. And so we're joined here today by three authors on this recent paper in Journal of Cellular Biology. And they are Frederik, Guillaume, and Michiel. So I'm just going to ask everybody to remember to put your comments and questions in the chat box as we go along. And then we will allow you to unmute yourselves, show your video at the end, and have a robust discussion with our co-authors here. So Guillaume, let me just hand this over to you to introduce the members of your team, please.
Yeah, well, members of my team.
Well, sorry, it's a team. I don't mean that in an authoritarian sense. As a group here. Yeah.
Okay, the group, the three musketeers of EVs. Yes. So it's my pleasure to introduce Frederik Verweij and Michiel Pegtel. So Fred was a PhD student in virology in the Cancer Research Center in the team of Michiel, and I had the pleasure to meet him at one of the ICEV meeting. I think it was in Rotterdam, where he presented his the fantastic tool he developed in the team of Michiel, and then I could convince him to come to my lab. So we can transpose that in zebrafish, but that's another story. So Fred did then a really productive postdoc with me, and is now rewarded by all this success as now a team leader position in Utrecht with his team of extracellular vesicle biology. He received prestigious grants like ERC recently, so he's really on good track. So I think, Michiel, we can be proud of our baby, big baby. So Fred, I'll let you the floor now.
All right, so I'll share my screen then. All right, so you should all be able to see my screen now.
Looks great.
Okay, perfect. So indeed, well, thanks Ken for the invitation to present this journal club. And so in this journal club, I want to show our latest paper that indeed was done. Well, actually, the oldest data stemming from this paper was still from 9 years ago at the Amsterdam UMC. Part of it was done at the Curie Institute, part at the Institut de Psychiatrie de Neurosciences in Paris, in Guillaume's lab also, and also the last part was also done in my lab here now at Utrecht University. So it's really a team effort and also a time-spending effort. So in this paper, we're actually studying the impact of dynamic membrane contact sites between the endoplasmic reticulum and late endosomes to see how they impact exosome secretion by actually allowing GTPase conversion on these vesicles, and I will explain this all in the following slides. So this indeed started, as Guillaume was already alluding to, in the lab of Michiel Pegtel, where we developed an optical reporter to visualize EV release, exosome release specifically. I think most of you will be familiar with this tool. But in brief, what we did was we used the tetraspendent CD63 that is highly enriched on a broad range of exosomes that we coupled to a pH-sensitive version of GFP. And since the pH in multivesicular bodies is generally low and the extracellular pH is generally high, you get a pH switch at the point that the MVB fuses with the plasma membrane. And this therefore results in a burst of fluorescence that you can observe by live cell microscopy, as you can see here. So for this, we most often use turf microscopy because that really allows you to specifically look at the plasma membrane part that is facing the cover slip. So you have a large surface area of plasma membrane that you can all visualize it at once. And there you see these bursts of fluorescence, as you can see here. And we then also did, that was in the lab of Guillaume, we did a correlative light electron microscopy to show that indeed all these fluorescent events were correlating to instances where multivesicular bodies, and here you see the limiting membrane, were fusing with the plasma membrane and releasing these small intraluminal vesicles into the extracellular space. So this gives you then a dynamic insight into exosome release specifically, and then allows you to use all kinds of different triggers, but also inhibitors, for instance, for snare proteins, to really decipher what signaling pathways can trigger exosome release and what proteins are involved in really this last step of the fusion of multivesicular bodies with the plasma membrane. And so this was published in 2018, also in the Journal of Cell Biology. But then we went on. And indeed, as Ken also mentioned, we really wanted to know the inner workings of exosome release. Because indeed, how we usually depict exosome release is that you have a multivesicular body that approaches the plasma membrane, fuses, and then you have your exosomes. Whereas in reality, of course, there are many more compartments in the cell, as you can see here, that all interact with each other. One result of that is that, of course, that exosome secretion in itself must be the end result of many intracellular interactions that happen inside the cell. And so what you can also see from this picture is that, in fact, there are multiple compartments in the cell that carry these intraluminal vesicles, and not just what we consider the canonical MVBs. So basically, we wanted to ask ourselves two questions. First of all, which of these compartments are actually the ones that are fusing with the plasma membrane? So meaning, amongst others, like at what stage do they fuse? How mature are they? Do they show catalytic activity already or not? Or is it more of an early stage of endosomes that fuse? And the second question that we want to answer was actually how the cell renders compartments fusion competent with the plasma membrane. So what steps are necessary to bring these multivesicular bodies to the plasma membrane in order for them to fuse, right? So, and of course, for this, we can now also use, again, the CD63 fluorine to specifically look at what happens at the plasma membrane. So this is what I've just shown you, but what we also did was to combine this with other proteins of interest tagged in a different color, in RFP, for instance. And this then allows you to also look at other proteins of interest to see, for instance, red GTPases. I will come back to that later, to see if they're also present at the site of fusion, meaning that they associate to these multivesicular bodies at the point that they fuse. So we can use CD63 fluorine, green pH sensitive version of CD63, or CD63 Fuji, or we have some different variants and different colors. And so we can also combine a red pH sensitive CD63 with a protein of interest tagged in GFP. And using this, you can then really define, do some molecular identity tracing of the compartments at the moment that they fuse with the plasma membrane. Right. So now I will just follow the figures. Since this is a journal club, I will just follow the figures from 1 till 6, I think, and just walk you through the figures. So the first question that we want to answer was, as I mentioned 2 slides back, at what stage do these compartments actually fuse with the plasma membrane? So how mature are they? And of course, as you know, like there are multivesicular bodies, there are late endosomes, there are endolysosomes, there are lysosomes. So how can we distinguish between those different compartments? And how can we distinguish them also from earlier compartments, like early endosomes? For this, we took initially 2 markers, EA1, that has classically been associated mostly with early endosomes, although we now know that they're also on a subtype of multivesicular bodies. And LAMP1, which has classically been associated mostly to lysosomes, however, also shows a lot of overlap with other CD63 compartments. So it's not exclusively lysosomes, it can also be endolysosomes and also, or even earlier states of late endosomes. So we use this dual color curve approach that I just showed you to first look at LAMP1. In this case, we use a red pH sensitive version of CD63. So you can see an event happening here. Over time, you can follow it. So you see it here present at the plasma membrane. And then if you look in the green channel, and this cell was transfected also with a LAMP1 tagged with GFP, you see that indeed, even prior to fusion, you see a weak spot in green here that co-localizes with this fusing multivesicular body. And so you can do that over multiple cells, multiple experiments, and then average the signal that you get, and then compare the two. So in B, you see the fluorescent intensity peak average over more than 15 events in different cells, different experiments. And this is a typical fluorescence peak, intensity peak that you see for CD63, so CD63, fluorine, or Fuji. And then if you look at the exact same spot by just drawing a circle at the fusion event and then measuring the other channel, you can see that indeed there's always a slow bump also of LAMP1. So LAMP1 is weakly present at the site of fusion. But if you look, for instance, at EA1, that's more in earlier endosomes, you see that this completely flatlines, meaning that indeed we're mostly looking at the later stage of endosomes that are CD63 and LAMP1 positive. What we did next was look at endogenous LAMP1 and CD63 labeling, and also overlap in the cell. And as I mentioned, usually CD63 and LAMP1, they perfectly overlap. So everywhere, all endosomes that have CD63 also have some LAMP1. So that's what you can see here. So the correlation is pretty high, 0.7. But you can also appreciate from this picture that there are some differences in intensity. So although CD63 and LAMP1 are almost always together, if you would look at the individual spots, you see that sometimes some endosomes have more endogenous CD63 than LAMP1, and sometimes they have more LAMP1 than CD63. Sometimes they coincide, but you see that here actually the R is much lower, it's 0.3. So the correlation between intensity in the compartments is quite different, and this might reflect, for instance, that the ones that are much more LAMP1 positive than CD63, that they are more lysosome or endolysosome-like than the ones that are more CD63, higher in CD63 and lower in LAMP1. So the presence of LAMP1 per se does not yet tell us whether we're looking at an earlier stage type of late endosome, or for instance already at late endosome that have fused with lysosomes, forming endolysosomes. So for that reason, we wanted to look further into this endolysosome formation process, and also how lysosomes are reformed from endolysosomes to form a pool of terminal storage lysosomes. So this is a process that has been studied also in the lab of Guillaume with Christine Bissig as the lead author, where they could show that indeed multivesicular bodies or multivesicular endosomes that fuse with terminal storage lysosomes form endolysosomes. And then by conversion from PI3P lipid to PI3-5P2, mediated by the enzyme called PIC5, you actually enable this reformation, so this budding off from the endolysosomes to form new terminal storage lysosomes that can then again fuse to multivesicular bodies. So you need this process to to allow content degradation of multivesicular bodies, but also to after of course everything is degraded, you need to recycle these degradative enzymes so that you allow a new round of fusion and degradation with new multivesicular bodies. So this process of lysosomal reformation critically depends on PIC5, which is also something that you can inhibit, as I will show you after this. So just to give you some more background on endolysosomes, as I mentioned, they fuse with and separate from lysosomes. They show proteolytic activity because they degrade the contents, but they also contain the lysosomal TRPM1 calcium channel. So we use this information actually to get more information about the compartments that fuse with the plasma membrane when we look with CD63 fluorine. And so when you inhibit this PIK5 enzyme, you actually inhibit also the lysosomal reformation. So this means that lysosomes keep fusing with multivesicular bodies, but there's no lysosomal reformation happening any longer, which means that they keep growing and growing, the endolysosomes, and at some point you deplete also your pool of lysosomes. So that's also what you see happening here. This is the control situation. In this case, we block this PIK5 enzyme. So you see these large swollen endolysosomes in the middle of the cell, and you can also see that actually quite immobile. So if you look here, the normal endolysosomes and late endosomes, they're quite mobile, but these large swollen endolysosomes, they're completely immobile. And here you're looking in the middle of the cell here as well, but if you look at the bottom part of the cell, you can actually distinguish still here. So in red, you have CD63 coupled to M-cherry. You can see actually that there are still quite some smaller compartments that are mostly CD63 positive, not so much LAMP1, that are still able to, that are still actually mobile. So it's mostly the ones that are high in both CD63 and LAMP1 that are, or LAMP1 alone high, that are affected by this drug, and not so much the smaller ones. All right. So what we then did was we looked at those cells treated with this PIK5 inhibitor to see if we would find a reduction in fusion activity with the plasma membrane, but we did not. So it seemed that inhibiting lysosomal reformation does not affect the ability of multivesicular bodies to fuse with the plasma membrane, which suggests that this might be in earlier states of compartments, so that the compartments that fuse with the plasma membrane are upstream of this lysosomal fusion, budding, and reformation, and then fusing again pathway, right? So when you look a bit further into that, and again separate LAMP1 and GFP, we also looked at catalytic activity using this magic red dye, which is a substrate for cathepsin B. And then if you look at the overlay between this catalytic activity and the different markers, you see that LAMP1 overlaps a lot with magic red, whereas CD63 fluorine overlaps to a much lower extent with magic red. And you see that even a bit better if you, again, induce this endolysosomal swelling with epilimod, where you can clearly see this catalytic activity contained inside these LAMP1 compartments, but not so much in CD63 compartments. So that's also quantified here. And so then the final part of this figure one, we also looked at overlap of magic red, because what you see here suggests, of course, as I mentioned, that the compartments that fuse with the plasma membrane have not yet encountered lysosomes, which suggests that compartments that fuse do not have degradative capacity. That's what we looked at with combining magic red directly with CD63 fluorine. And indeed, at the places where you see a fusion event happening, you don't see any magic red, whereas you can actually see a lot of degradative compartments, so catalytically active compartments closer to the plasma membrane, but they are not the ones that are fusing, right? Same for this lysosomal calcium channel that I mentioned, TRPM1. So here again, you see this fusion event, but there's no TRPM1 present on this place on the same spot that you can also see quantified here. So this really suggests that the compartments that fuse are at an earlier stage, non-degradative. And then to also exclude it, maybe it had to do anything with autophagosomes. We also did the same thing, but using LC3B M-cherry. And also here we found LC3. We did not find any LC3 signal at the site of fusion. Right. So as a summary of this first figure, what we've shown using this approach is that the compartments that fuse with the plasma membrane under steady state conditions, so the most typical contributors to exosome release are actually these canonical MVBs that contain LAMP1 and CD63, but no catalytic activity or also no LC3. So this tells you more about the maturation stage. But one other way to look at endosomal transport and also maturation is actually by looking at GTPases. Now, over the past years, even decades, people have implicated different rep GTPases and small GTPases in the exosome release pathway. That's what you can see here. It's a nice slide that I got from Clotilde Thierry, where you see these different reps, for instance, 11 and 35, but also rep 7 and rep 27A and B being implicated in exosome release. And what we wanted to ask ourselves is if these are all different separate pathways from each other, or if they are somehow also interrelated. So if we could get some more insight of what is actually happening inside the cell. Right, and again, for this, of course, using this duocolor turf approach is a very nice way of seeing what rep GTPases associate to compartments at the point that they fuse with the plasma membrane. So what we did then, this is figure 2, start of figure 2, is that we first looked inside the cell using normal confocal microscopy. So here you get like a snapshot, a general overview of all the compartments or most of the compartments that you have in the cell, mostly in the middle of the cell. So we looked at rep 5, rep 11, 7, 8B, 27A, and 27B, and looked at the overlap of these rep GTPases or small GTPases with CD63. So I will show the quantification of this in two slides after. But hopefully you can already appreciate from this figure here that the rep GTPases that associate most with CD63 are rep 7 and the small GTPases are 8B, where you see a strong overlap and very low overlap with, for instance, rep 5 or 11 or 35. All right, so here we're looking in the middle of the cell at basically the total pool of endosomes. But if we now look turf microscopy, duocolor turf microscopy, you can specifically look at the endosomes that are very close to the plasma membrane, and in particular the ones that are fusing, and see which of the reps that I just saw that we were just looking at are associating with compartments that at the moment that they fuse with the plasma membrane. If we then look at rep 7, to our surprise, we did not see any rep 7 at multivesicular bodies that were fusing with the plasma membrane. So you see that there's absolutely no trace of rep 7 near this fusion event. For rep 5 is the same, but that's of course logical because it's a more early endosomal marker. We also did not find any rep 11, no R8B. We did find, however, some rep 27A and also some rep 27B. All right, so as I mentioned, the quantification. So if you put this next to each other and you look at the larger population of endosomes throughout the cell, you see actually that rep 7 and R8B are most strongly associating to CD63. But if you look specifically at the pool that is fusing with the plasma membrane, actually it's mostly rep 27A and B and not rep 7 that is associating to these compartments. However, and also, I mean, I've just shown that in one of the previous slides, we do know that somehow rep 7 is involved in fusion with the exosome release pathway. Also shown here in this experiment where we knock down rep 7, and indeed here you see a very strong decrease in fusion activity. So if you interfere with rep 7, you get much less multivesicular bodies that fuse with the plasma membrane. So how does it work? So how is rep 7 involved? Well, in fact, rep 7, so I mentioned they are important for maturation and transport. So one of the functions, one of the ways how they do it, so one of the effectors of rep 7 are proteins that we call membrane contact site mediators. And membrane contact sites are very close appositions of compartments in the cell, in this case, late endosomes and the endoplasmic reticulum, that are so they're in very close proximity, but they're not fusing. And many of these memory contact sites mediators, they interact with ER resident protein VEP-A. And those include, amongst others, protrudin and ORP1L. So because of this involvement of Rep7, suggesting the involvement of Rep7 upstream of the fusion, because we did not see it at the site of fusion, we were interested to look if these memory contact sites also are playing a role in the modulation of exosome release. So to start, we first looked at protrudin and ORP1L to see if they were also impacting exosome release. So that's something that you see here, figure 3, we're now, figure 3a, so Rep7a I've already shown, depletion causes a large reduction in exosome release, same for interfering with protrudin, and also ORP1L, so those two memory contact site mediators, if you deplete them, you get a very stark reduction in fusion activity. So first, we looked a bit further into protrudin. So this picture just shows the localization of these compartments. There's, well, at least in our case, this is a 5-4A, a mutant that's not able to interact anymore with late endosomes. So if you overexpress the wild type of protrudin, you can indeed see an increase in fusion activity, but if you overexpress a mutant of protrudin that is not able to interact with late endosomes, you don't see this increase any longer. But since this protein was already described in 2015 by Camilla Ryborg et al., we decided to focus a bit more on ORP1L. And ORP1L is actually an interesting protein because it has 2 different conformations. So in one conformation in cholesterol low conditions, it actually forms these memory contact sites by interacting with this FFAT domain to VEP-A. However, under high cholesterol conditions, you have this cholesterol sensing domain of ORP1L here that senses this cholesterol and then closes this conformation that allows for the dynein-diactin recruitment, and which has as a consequence late endosomal repositioning towards the minus end of the microtubules. Right, and so since this process is dependent on cholesterol levels, we first looked a bit further into this by using a drug, a chemical drug called the U drug, which actually increases the late endosomal cholesterol levels. So also increases this perinuclear clustering of late endosomes, as you can see here. So this is the control situation. Here's the U drug situation. Here you see all these endosomes very close to the nucleus. And as a control for this, so of course we were looking here at fusion activity, comparing that into both situations, but as a control for this, we also looked to 2 other types of secretory vesicles. So the first one was neuropeptide Y as a marker for soluble secretion, NPY. And the other one was VAMP2, which is a marker for well, plasma membrane deposition of transmembrane cargo. And so when you do that and compare the wild type or the control situation, sorry, versus the UDREX edit condition, you see that VAMP2 and NPY, they do not differ whether or not you add this cholesterol interfering drug. So the fusion activity stays the same. Whereas for CD63, we see a very strong decrease. So if we move these late endosomes away from the plasma membrane towards a perinuclear area, we cluster them there, we see a stark decrease in fusion activity, suggesting again that indeed this ORP1L protein is important for maturing and probably also bringing the late endosomes towards the plasma membrane to fuse. One other thing that we checked was then also to see if this U drug indeed, as suggested by this picture, removes multiple circular bodies, late endosomes, away from the plasma membrane more towards the periphery. And indeed, by using this turf microscopy, since there we're just looking at the compartments closer to the plasma membrane, we could quantify the number with and without U-drug, and indeed found that there was a stark decrease in multivesicular bodies close to the plasma membrane. So we then decided to look further into this ORP1L protein, for which we also used two different mutants to mimic those two different conformations. So the open conformation can be mimicked by removing this cholesterol sensing domain, which is called the ORD domain. In this case, you always force membrane contact sites, as you can see here. And this this closed conformation can be mimicked by replacing this cholesterol sensing domain with a PhD sequence, which always closes this protein conformationally and always recruits this dynein-dynectin motor, and therefore also causes a slightly similar relocalization of compartments towards the perinuclear region, right? So in figure 4, we were then looking further into this, how ORP1L was behaving and affecting exosome release. So first we looked at the late endosomal localization, and indeed we found that, so this is the normal localization of CD63, so it's mostly perinuclear, but also a bit scattered through the cytoplasm. It shows much more scattering if you delete this ORD domain and force these memory contact sites, whereas if you use the mutant, overexpress the mutant that forces this dynein-dynectin recruitment and brings the late endosomes towards the nucleus, you see this indeed, this large clustering, all these kind of grape-like structures of late endosomes near to the perinuclear area. All right, so then we wanted first to confirm, and this was done by Maarten Bebelman in the lab of Guillaume, to confirm that indeed this delta ORT domain, the delta ORT mutant was indeed forcing these membrane contact sites. So for this we did a Clemon section. So here you see it for the wild type situation. You have your EM picture here with an overlay of the fluorescent signal. So you can really zoom in on this endosome here. But you see here nicely tubulating. You see these interluminal vesicles with basically no ER around, or at least it's not directly attaching to the late endosome. Whereas in this, while you overexpress this delta ORT mutant that forces memory contact sites, you can see also a nice multivesicular body, but now in this case, completely enwrapped by endoplasmic reticulum here in orange. So indeed, these mutants are able to mimic these two situations. And then, of course, we were interested in how this was affecting endosomal maturation, but also fusion activity. And what we found to our surprise was that if you force these membrane contact sites, is what you see here. And if you then look at the overlap between LAMP1 and CD63 and compare that to the situation where you basically inhibit memory contact sites and force this perinuclear aggregation of late endosomes, you see actually an increase of CD63 LAMP1 overlap. So compared to the wild type situation, if you force memory contact sites, you get a lower amount of LAMP1 CD63 overlap. But if you force the perinuclear aggregation of these compartments using the delta ORT PhD mutant, you see a large increase in overlap between CD63 and LAMP1. Right, so then we looked at the fusion activity, and interestingly, in this case, we found that if you force, so continuously have these memory contact sites, you completely enwrap them, you see a stark decrease in fusion activity, whereas if you continuously recruit this motor that brings it towards the nucleus, actually this result, the end result is that you get an increase in multivesicular bodies that fuse with the plasma membrane. Right, so of course we also wanted to confirm this by the golden standard, let's say, using EVs isolated from the supernatant by ultracentrifugation and then doing Western blotting, where you see indeed the same trend, so a decrease of CD63 levels in the memory contact sites, forcing condition, and an increase in case you force the recruitment of this motor and the perinuclear localization of these compartments. And of course, as you can imagine, looking at this compartment that's completely enwrapped by the ER, this would suggest indeed that also something is happening with the motility. And that's indeed what we found. So if you look here and you compare the C63 compartments that have ORB1L delta ORT recruited, so have these forced memory contact sites, and you compare them to compartments that are green alone, so meaning that they do not make these memory contact sites, likely or potentially by the endogenous ORB1L, but in this case it's not forced. So if you compare the two, you can see that the median speed of these compartments, the relative velocity, is much lower in the case that we force these memory contact sites on these compartments, which probably partially explains why we see this decrease, this drop in fusion activity. But then interestingly, because of course this could suggest that in one way that memory contact sites are perhaps inhibitory for fusion activity, so for exosome release. However, to test this, we used another mutant of ORB1L, which does not have this forced motor recruitment, but in fact just misses the FFAT domain that it needs to bind to VEP, so to form these memory contact sites. So this mutant does not force the recruitment of the motor. It does still allow it. It's only the only thing that it cannot do is form these memory contact sites. And to our surprise, what we saw here was also again a bit consistent with the complete depletion of ORB1L, was a stark decrease in fusion activity. Right. So altogether, this data suggests that you really need a dynamic ORB1L mediated memory contact sites with the between the multivesicular body and perivascular reticulum for proper endosome maturation, but also transport. So you cannot have either one or the other. So yeah, basically you cannot live with them or without them. You need memory contact sites, but you need to have them in a dynamic manner in order to ensure proper maturation and transport of these compartments. Right. And then what we did, which is also relevant for the next slide, is we also overexpressed some of the other mediators. So RILP1, which also causes the relocalization of these compartments towards the nucleus, and SCIP1, which goes in the other direction. And to our surprise, actually, in both cases, we could see an increase of fusion activity. So that's also shown over here again. So we've looked at ORB1L, that's in this complex via REP7, but also with RILP1. So if we overexpress this, this protein, this RILP1 protein, we actually also stimulate movement towards the nucleus, as I just showed you. So you see this increase, and if we inhibit this motor complex assembly, you get a decrease. So this is a bit puzzling, right? Because now it seems that if we send late endosomes towards the nucleus, so away from the plasma membrane, somehow we actually we stimulate exosome secretion, and the plasma membrane is on the other side. So how does that work? One other protein that we looked at was SCIP1, as I also just shown. So this is more logical. So if you overexpress SCIP1, you induce the peripheral relocalization of late endosomes towards the plasma membrane, and in this case, indeed, we increase fusion activity. And if you inhibit this protein, R8b, which is also important for the relocalization towards the plasma membrane, you see a decrease. So it means that for sure moving towards the plasma membrane is important, but for some reason also moving late endosomes towards the nucleus is beneficial for exosome release. And then of course we finally have this RET27 that I've also shown you earlier, that has been implicated in the final stages of exosome release. So we were wondering how this would happen, and how to explain these slightly counterintuitive results of sending late endosomes one way, stimulate exosome release, sending late endosomes the other way, also stimulating exosome release. So how does it work? First, we thought that, how is that possible? Is there some sort of a tug of war happening? But then, while we were working on this project, this paper by the group of Jacques Nevis in Leiden came out showing that there's actually a switch between Rep7 and R8B that is mediated by this protein called TBC1B15, suggesting that actually there's no tug of war happening, but you first have Rep7 that relocalizes late endosomes towards the nucleus. That's then followed by a switch to R8B, which then transports late endosomes towards the plasma membrane, and then switches to Rep27 to complete the fusion. Right? So to test this, we first validated if indeed Rep27A wild-type overexpression could increase exosome release and dominant negative overexpression could decrease it, and that's indeed true, as you can see here. And this, of course, also suggests that if you look at if this really is the step, so first Rep7, then R8B, then Rep27, then of course you would expect that there's much more overlap between R8B and Rep27 than there is between Rep7 and Rep27. Right? So that's what we tested here in the figure 5, B and C, where we co-express 27A and Rep7A, and you see indeed there are some instances of overlap, but there are not that many. Whereas if you look at R8B and Rep27, you see actually a lot of R8B positive compartments that are decorated by Rep27A. So and if you quantify that, indeed, you see that there's quite a difference. Well, this I've already shown. So R8B is also important for the fusion activity. So that makes sense now. And one other thing that we observed was that if you overexpress Rep27 in combination with either Rep5 or Rep7A or R8B, only in case that we co-expressed R8B in Rep27, we saw all of a sudden a lot of these compartments that were decorated with Rep27. And indeed, when you quantify that, you see that there's a huge increase in Rep27 positive endosomes if you overexpress R8B. And also that the intensity of Rep27 on these endosomes is also the strongest in case you overexpress R8B and not, for instance, Rep5 or Rep7. Right, so this suggests indeed that we have this double GTPase switch movement that's necessary to reach the plasma membrane. So then the final question that we want to answer was that if to see if indeed ORP1L was necessary for this Rep7 to R8B switch and then also to the Rep27A switch. So this would suggest, of course, that if you interfere with ORP1L, use the mutants or you deplete it, this will also affect switch to R8B, but also to finally to Rep27A. So and this is the second part of figure 5. So if you do that for Rep5A and you use the different mutants and the wild-type version of ORP1L, you don't really see a difference. You actually take the two populations actually further apart. So that means that when you send endosomes to the perinuclear area, you have less overlap with Rep5. Rep7, you see that there's always a very high overlap between Rep7 and ORP1L, which is logical because ORP1L is an effector of Rep7. And now, interestingly, when you look at R8B, you see that in case you form these membrane contact sites and you, so you have a more scattered cloud of endosomes, you actually decrease the overlap between R8B and ORP1L, whereas if you, and also Rep7, because they always overlap very well, but if you use this mutant that stimulates exosome release, the delta or the PHZ mutant, you actually increase the overlap between Rep7, ORP1L, and R8B. All right. So then finally, to make the circle round, so what we first did here was, because this last experiment here suggests that In this case, you indeed stimulate this R8B. In case of delta ORT PSC overexpression, you stimulate exosome release because you get more R8B recruitment. So here what we did was we compared this increased situation for the delta ORT PSC with delta ORT PSC overexpression in combination with overexpression of a dominant negative version of R8B. In that case, you expect, of course, that you don't see this increase anymore, and indeed, fusion activity in this case was back to basal levels. And then finally, what we did was we looked at the effect of organelle depletion on CD63 and Rep27 overlap. So organelle is here, and you have those two steps, 7 to 8B, 8B to 27. You do expect that interfering with organelle ultimately also has an effect on the Rep27 recruitment. And that's indeed what we could see here. All right, so this leads to the final concluding slide. So what we've shown is that indeed early endosomes, after their switch from Rep5 to 7A, they move towards the nucleus in this dynein-mediated movement, where they undergo these dynamic memory contact sites with the ER via organelle, which is necessary for their maturation and transport. Then in this perinuclear area, they undergo the switch from Rep7 to R8B, which then allows the multivesicular bodies to exit from the perinuclear cloud and move towards the periphery, where they can then switch from R8B to Rep27, which then really primes the late endosomes for fusion with the plasma membrane. All right, so very recently, this paper came out from Ganesh Shalka in the lab of Juan Bonifacino, where they actually showed that R8B and R8A actually not only are important, well, what they show is actually that if you interfere with those proteins, you also inhibit the fusion of lysosome, late endosomes and lysosomes. So what it now suggests is that R8B is important for this step, but the combination perhaps of R8A and R8B is also affecting or promoting this fusion with lysosomes. So this suggests that R8B per se is not the one that in the end stimulates fusion with the plasma membrane, but that only at the point that you have Rep27 on your compartment, you really have primed your multivesicular body, your late endosome, to fuse with the plasma membrane and release exosomes. All right, so with that, I'd like to thank all the people on this slide, so all the sources of funding, but also all the sources of fun, which are the people that I've worked with. And of course, to thank you for your attention, and I'm happy to take your questions.
Thank you so much, Fred. We are now going to allow everybody to unmute themselves who would like to ask questions. And just a reminder that if you didn't hear at the beginning, please put your question in the chat box or your comments, and then I will get to you probably in order. We'll just go through here in order. So please be ready to turn on your mic and engage with our speaker and his co-authors. All right, so let's get started here with Devika. Devika, you have a question here about CD63. I just want to point out that that exosomes and small EVs are not always the same thing. So small EVs can also come from the plasma membrane. So just because we see something that's less than 100 nanometers in diameter doesn't necessarily mean that it came from the endosome. So, but please go ahead with your question.
Thank you. Thanks for that clarification on what constitutes as small EV. So my question is, I've seen, I think, about a paper or two suggesting that CD63 is differentially expressed both by exosomes as well as the plasma membrane derived ectosomes, or sometimes referred to as microvesicles. So in this case, if that is indeed the case, can CD63 be exclusively used as a marker for exosomes?
Yeah, so that's a good question. I think the answer is then no, although they have shown an enrichment, but of course it doesn't mean that it's really a marker that you can use to exclude one population from the other. It just shows that it's enriched in smaller EVs that indeed are not all exosomes. What you can do, however, in our case, because we are looking with live microscopy, is see the instances where late endosomes fuse with the plasma membrane, which, as we've shown by the correlative light electron microscopy, causes the release of ILVs. And then in that case, you know for sure that exosomes are released at that point. But after that, after you isolate vesicles from the supernatant and then look at CD63, of course, at that point, you cannot tell anymore whether it's an ectosome or an exosome.
Thank you. If I could just go back to Ken, your comment for a quick second here. So I agree with you that not everything that is smaller than 100 nanometers will be an exosome. So in that case, if this is a smaller EV released from, say, from the plasma membrane, what would be, well, I guess, you know, I guess an appropriate terminology will still be to use based on the size, right? Small versus large, right? No, it's okay. Thank you.
All right, let's go then to Ananga Ghosh. Question about RAB27A.
Hi, Fred.
Hi.
So, initially in figure 2, you have talked about and in your chart microscopy, it shows that there is not much of a 27 plus vesicles and 63 was positive.
I couldn't.
Yeah, Ananga, I'm sorry. I think your microphone is not letting us hear you very well. So I'm just going to read the question that you have in the chat box here. So RAB27A is a known positive regulator of exosome biogenesis. I was wondering why there was not much overlap between CD63 positive and RAB27A positive EVs.
Yeah, that's a very interesting question. Of course, it could mean different things. It could indicate that maybe a large population of late endosomes in the cell is not meant for secretion. But it could also mean that as soon as RAB27A is on your compartment, fusion happens really fast. So that they're really, it's, as we've shown, it's really, we think it's really one of the last, really one of the final steps. So as soon as it's on there, you trigger the docking and the fusion. So that's maybe why there's not that much RAB27 on these compartments.
Okay, thank you. Thanks a lot.
And Anna, could you please follow up with what you meant about RAB27B? So Fred, maybe so Anna says here, as compared with RAB27B and confocal or turf at the site of fusion. So could you contrast the findings on 27A versus 27B?
Yeah, so we've followed up more on RAB27A, and we did find both RAB27A and RAB27B at the site of fusion. It seems the paper from the group of Clotilde Thierry with Matthias Oschowski as lead author suggested that RAB27A is perhaps the later acting RAB27 variant. We followed up mostly with RAB27A and did not really go into RAB27B that much. But still, of course, it would be interesting. There are some suggestions that they work in parallel, and there are some suggestions that say that they work consecutively, so that you have first RAB27B recruitment. Also, if you look at effectors of the two isoforms, it's rather RAB27A that can really interact with the actin in the myosin. So it would make sense that RAB27A is a bit more later acting. However, they could also have redundant functions.
Excellent. All right. Next question from Ganesh Shalka. Ganesh, please go ahead.
Hey, Frederik. Amazing talk, as always. So my question is regarding the turf analysis that you conduct in a cell. So in the turf field, you have peripheral area, and then you have an area next to perinuclear site, right? So when you image the event, do you have have you seen the differences in the fusion event that occurs next to the nucleus and the peripheral population?
In that sense, peripheral. Yeah. So when I mention peripheral, I usually mean that just looking at the cell, this is here, periphery is here.
Okay.
But of course, it also,
Yeah.
you can also say plasma membrane close to the nucleus and plasma membrane further away from the nucleus. So we haven't yet looked a lot into the location of these events. Also because most of our cells are not migrating that much. Okay. But of course, it could be interesting to look more at polarized cells and see if there's indeed a preferential area of secretion. So with TURF, indeed, we cannot really distinguish, we do not distinguish between plasma membrane close to the nucleus and plasma membrane further away from the nucleus.
Okay. So basically, so when you count those events, those are like a cumulative events, and you cannot say with surety that whether they are perinuclear versus peripheral. So at this time point, I guess. Yeah,
we can use, like, if of course if you co-label the nucleus, you could, but we haven't done it so far.
Okay. All right. Thank you so much. Great work.
Thanks for the question, Ganesh. So we have proteins on EVs, but there's also other components. Esther Naltatun.
Yeah, thanks, Fred, for a very nice talk. I was wondering, since you see a role for cholesterol in the membrane contact site formation, may this also explain the very old published data of about 20 years ago, where people on EM pictures saw cholesterol-rich and poor multivesicular bodies. And that's really heterogeneity of multivesicular bodies. And I think they even claimed that mostly the cholesterol-rich ones were the ones that kind of were prone to release the vesicles, and there was also cholesterol in the vesicles. So are you somehow able to relate those very old data to what you are seeing now?
Yeah, that's a very interesting question. Indeed, there's this, I think maybe it's this paper where they showed cholesterol-rich MVBs fusing with the plasma membrane. And since it's a bit hard to catch this with electron microscopy, of course, it suggests that maybe there's a lot of cholesterol-containing multivesicular bodies that fuse with the plasma membrane. So our data with the U-drug suggests the opposite, because we move the cholesterol-rich late endosomes towards the perinuclear area, and they seem to be much more immobile there than compared, for instance, to the delta or PhD cluster that you see there. I have like one remark that you have to take into account there is that you're probably doing a lot more than just accumulating cholesterol. So it's affecting MPC1. And in case we use the delta or PhD, we actually relocate the vesicles irrespective of the cholesterol content. There are also one other remarks that there's some different papers, one looking more at virus release, where they see, for instance, that the U-drug decreases it, but I think that there's another paper where they showed an increase. So this could also depend a lot on timing. So maybe usually we do a relatively short treatment of the U-drug. Maybe over time, when you really start to accumulate these compartments and perhaps also inhibit degradation, at some point the cell needs to get rid of it, and then they fuse with the plasma membrane. And one final remark is, of course, that, well, what we've also shown is that, so with the delta or PhD, you localize these compartments to the perinuclear area, but there they undergo the switch from Rep7 to R8b. And since ORB1L is an effector of Rep7, this means that they also at some point lose ORB1L. So over a longer time period, you actually will lose it. So it could still be compatible with cholesterol-rich multivesicular bodies fusing with the plasma membrane. But I think that maybe also, Guillaume, since you have a very nice ongoing story in the lab with Roberta also, maybe you can also comment on that.
Yeah, thank you, Fred. Indeed, I think there is two points to take into account. First one is the localization of this cholesterol, whether it is on the limiting membrane, and so it can recruit effectors like RABs, or whether it's enriched in intraluminal vesicles and no longer on the limiting membrane. So what Wipke Mobius has shown is that the MVB that secrete are enriched with cholesterol on the intraluminal vesicles. So maybe there is already a sorting step of cholesterol to the intraluminal vesicles, and so that the cholesterol is depleted from the limiting membrane. And the second thing is also to take into account how we can sort this cholesterol to the intraluminal vesicles. And the paper that we have in preparation with Roberta Pamuli is showing notably that it's the role of CD63 to sort this cholesterol to intraluminal vesicle. So maybe we are kind of closing a loop here. We're using the CD63 fluorine. We have finally the perfect tool to act on cholesterol and maybe to boost the secretion of exosomes, so the fusion of MVBs. And I think that the ongoing work of Michiel with Martin Webelman and Fred is also kind of showing that, is that by using this CD63 overexpression, we may also alter secretion, and that may deal also with the sorting of cholesterol to MVBs.
But in relation to that, so in your picture, Fred, you show actually that most of the intraluminal vesicles are already formed before they reach where they form the contact sites. But is that kind of known? Do you know that that is the case, or may there also be intraluminal vesicle formation at a later stage, maybe even during contact with the ER?
Yeah, yeah, that's also an interesting point. So for sure, it happens much before, even before late endosomes form. So already in early endosomes and recycling endosomes, sorting endosomes, we see these intraluminal vesicles appearing. There are also some studies, in particular related to I think it's by Claire Footer's group, looking at EGFR that is stimulated and then internalized, and there's also late endosome compartments, and then she also implicated the mechanism via memory contact sites that might induce the ILV formation. So I do indeed think that memory contact site might also impact exosome, well, ILV formation, and as a corollary also exosome formation. Yeah.
Thanks, that's great.
Great. Michiel, did you have anything to add to that discussion?
Not necessarily of this discussion, but I was intrigued by the recent paper that shows that there's some some discrepancy in the results. But I don't know if the first author of that paper is still present, or.
Yeah, that's Ganesh.
Ganesh?
Yeah, hi, I'm here.
Hi, yeah, I really enjoyed the paper. And Frederik had some kind of explanation why the ARL8AB might be a little bit different. Do you have a similar explanation, or do you have maybe a different explanation on this discrepancy?
So I think the function of ARL8 and ARL8B, in our lab, we are trying to understand that, like why they functionally differ. So, and they have the ability to carry different cargoes, different membrane structures, ARL8A and ARL8B. And we see a phenotype when we knock out both of the ARL8 in the experiments. So I think that is, and that phenotype is more like a fusion defects and Hobbs complex and things like that. But when we try to understand the function of ARL8A and ARL8B in polarized cells specifically, we see they function differently. So I think the discrepancy is probably due to the since you knock down one of the gene, and we see the phenotype in double knockouts. So I think that's a majority of the reason.
Yeah. Because did you also apply the single knockouts then?
No, we haven't tried that because in those experiments when we do, the clustering phenotype is pretty mild. published in previous papers. So there's a possibility that, and we know that from our experiments, that RLate A and RLate B carry different size vesicles as well. So we are still characterizing them. That brings structures to peripheral site or to the.
Okay, because actually that was going to be my follow-up question. That if it seems that we have different routes and we get vesicle release, if there is also different in cargo, or in the end it doesn't really matter which route they take, the cargo is the same. And so, but you're looking at that, you said.
Yeah. So we are trying to address this question probably in polarized cells, because we can understand that, because HeLa cells and other non-polarized cells is difficult to study these questions. And in some of the cells, we can see this distribution of different RLate, and of course they contain multiple effectors. So in our paper, we actually don't know yet that what brings these vesicles outside to the plasma membrane. So that is not answered yet, but yeah, that is one reason thing that we're going on.
Okay, great. That was my question. Thank you very much. Thank you.
Yeah, thanks a lot.
Yeah, thanks for that. So we still have a few questions in the chat box. Our presenters today, are you able to stay on for another 5 minutes or so? Is that all right?
Yes.
Yes.
Okay. We'll try to get through this, but I think I'm going to ask these questions myself just to get this going rapidly. So the question here is there a universal marker for all small EVs or for all exosomes? My understanding is that we don't really have that yet, and that it may be that we'd have to look at multiple markers on the same vesicle to really get a good idea. Is that also your conclusion here? Or am I completely wrong, which I very well might be?
Yeah. No, at least as far as I know, indeed, well, there are single, double, and triple tetraspanin positive EVs, and even EVs that are negative for the CD63, 81, CD9 trits. And I don't think that, like, there's probably an enrichment for one or the other type, but it's not exclusive. No.
Indeed, depends on the cell types.
Also, yeah.
Cell type conditions. Yeah, we can have some differences there. So the next question here is about the pH sensitive dye. So Jinja Ju asks, it looks like there is some signal in the cell. How do you know if it's in the cell or associated with the membrane?
Let's see.
And unfortunately, Jinja does not have a microphone, so we can't ask for a clarification there.
Yeah, so we do indeed think that what we see when a fusion event is happening is a combination of cargo that is released. That's also what we've shown in the 2018 paper, but also CD63 fluorine that is in a limiting membrane that is then deposited in the plasma membrane. So it's a combination of the two. So if that, I don't know if that answered your question, but if it's more about how do you know it's inside or fusing, that's really the pH shift that is causing the increase in fluorescence, and that really happens in in milliseconds. So that cannot be a neutral vesicle that comes close to the plasma membrane and then moves away. So really, this fusion event itself it's relatively easy to spot it by the stark increase.
Yeah, so it's that bright.
It's not a dye. It's not a dye.
Yeah, maybe it relates to turf microscopy also. Turf microscopy is meant to see 200, 300 nanometer from the surface. So what you see is mainly the plasma membrane. So if you have a burst in your focus of the turf, that means that you are at the plasma membrane and not very deep within the cell.
Good. I think that's great. That should be clear. And last call, folks. And right now we have Manuel Varas who's asking a question. Manuel, I'm going to ask you to go ahead because I have, I think I saw a couple of questions, so maybe you revised this. So please go ahead so that I don't mess this up.
Hi, guys.
Hi. Hi, Manuel.
Hi, Big. Hi, Frederik. Hi, Guillaume. Hi, Kenneth. Thanks for the super nice presentation. I love it. And I have a question about if you know what happened with this contact between multivesicular endosome endoplasmatic reticulum when you have change in the level of RAP7 and RAP27, for example, in chemotherapy. Because we saw in one of our work that chemotherapy changed the level of RAP7 and RAP27.
Yeah, that's an interesting point. We ourselves have looked at more directly at the memory contact site mediators. Because of course, RAP7 can still interact with a lot of different proteins, also a lot of different memory contact site mediators. So yeah, but it could definitely. So we do see some changes in certain cancers that some of these memory contact site mediators are upregulated or downregulated. We have not studied yet the functional consequence of that. But if your RAP7 levels
in our case, yes, our RAP7 levels drop and RAP27 go high.
Yeah. Yeah, so for now I can just speculate, but I would think that having more RAP27 will increase fusion with the plasma membrane. So you have more compartments fusing with the plasma membrane, perhaps also at an earlier stage. So maybe.
But my point goes more than if you do you know if what happened with the localization of the multivesicular endosome, if it's close to the reticulum maybe or
maybe
not.
you have to consider what is upstream and downstream of a membrane contact site. The RAP7 is recruiting protein involved in the formation of a membrane contact site. From what I know, RAP27 is not. So I would say that if you decrease RAP7, maybe you decrease also membrane contact site, but it remains to know whether you're still recruiting RAP27 to MVBs to make them secretory.
Yeah, I understand. Yeah, thank you.
Yeah, but I would look at late endosomal localization in that case, if you can do immunostaining for that.
Perfect.
See if it changes. Yeah.
Super. Thank you very much.
Yeah.
All right. Well, thanks to everybody today for joining and for the nice discussion, and a special thanks to Fred and Guillaume and Michiel for this work that they've done. I think it's very valuable, and we look forward to where this is going to take us as well. And so, Fred, it's also great to see how your lab has come together and you're putting out such great work. So keep it up. And we hope to see everybody at a future EV club. So thanks again and have a good rest of the week. Take care, everybody.